We would like to introduce you to our bioadhesive chitosan hydrogel for the prevention of excessive blood loss in wounds. We found the area of wound repair to be a very interesting area of study. Many different bandages, glues, and dressings have been formulated, but as always, improvements can be made. We decided to take a look at the XStat 30 wound dressing, which is an expandable, multi-sponge dressing used to control severe, life-threatening bleeding from wounds in areas that a tourniquet cannot be placed. Now, according to the United States Army Institute of Surgical Research, 30 to 40% of civilian deaths by traumatic injury are the result of hemorrhaging. Of these deaths, 33 to 56% occur before the patient even reaches a hospital. The XStat 30 wound dressing can be used for up to 4 hours, which allows time for the patient to receive surgical care. The XStat 30 wound dressing is a device that is available in packages of 1 or 3 syringe-style applicators containing 92 compressed cellulose sponges that have an absorbent coating. These sponges expand and swell to fill the wound cavity, creating a temporary physical barrier to blood flow, while also increasing the pressure within the wound site to help stop bleeding. Each applicator can absorb approximately a pint of blood, and up to 3 applicators may be used on each patient. Now, within each sponge, there will be an X-ray detectable marker, which allows for the removal process once the patient is seeking medical care. Now, if you take a look at the image on the left, we see the syringe that's filled with a compact 92 sponges that will actually be injected into the wound site. The image on the right, on the other hand, shows the process and how once in contact with the blood, the sponges will rapidly expand to fill the cavity. This will increase the pressure within the cavity to stop the bleeding compared to many other applications which apply pressure to the outside of the wound. We would now like to show you this video on how the XStat 30 wound dressing really works within gunshot wounds.
Every drop of blood on the battlefield is precious. The faster you can stop the bleeding, the higher probability that you'll save that guy's life. There's always the common theme with the medics, improving technology.
XStat is a first-in-kind medical device designed to stop bleeding from small, narrow entrance wounds like a gunshot wound. You take the device, you literally plunge it into the wound or the bullet hole, and you shoot the sponges into the wound. As soon as they come into contact with blood, they rapidly expand and fill up the wound and compress it and get the bleeding to stop.
With the current standard on the battlefield, taking 3 to 5 minutes to pack a wound, soldiers lose a tremendous amount of blood.
One of the real innovations behind the XStat is that it's self-compressing, meaning that it expands from within the wound. What you normally have to do to get the bleeding to stop with a traditional dressing is apply compression from outside the wound and hope that that force or that compression gets to the site of bleeding. What XStat does is it expands in the wound and compresses outward, so it makes sure it gets all the potential sites of bleeding in the wound. And it works within seconds. And because it's compressing the wound, the medic doesn't have to, and essentially the treatment's done at that point.
All my friends are still on active duty, they're still deploying to combat. What drives me is to continue to make new products to help them on the battlefield that, from my own personal experience, I think are better products and will save lives on the battlefield.
The XStat 30 wound dressing proved to be very promising, and indeed it saved thousands of lives. With much research though, we found that there are problems with the application and materials of the dressing and the applicator. Each sponge contains an X-ray sensor which allows for the removal process. Therefore, when the patient comes into surgery, the surgeon must first locate each individual sponge, which is numbering from 92 to up to 276 sponges if 3 syringes were used. It is absolutely essential that each and every sponge is removed from the wound site. When this removal process is occurring, you're just reopening the wound site and aggravating the wound. This leads to a higher susceptibility to infections, damage, and hemorrhage. The process is also quite time consuming. Many of these patients do not have this time just to remove the sponges before seeking the actual treatment or surgery. During this removal process, the patient may experience even more blood loss than what they already have lost. Another downside to the XSTAT 30 application is that it cannot be used in abdominal wounds, wounds above the collarbone, or anywhere that a tourniquet cannot be applied according to the FDA. Taking all of this into consideration, we decided that it is highly necessary to develop a material that can save even more lives more efficiently, effectively, and safer. This led us to developing a bioadhesive chitosan hydrogel for the prevention of excessive blood loss in all sorts of wounds, big and small. A similar application using hydrogels is seen in the prevention of blood and air loss from lung fistulas. These lung fistulas prove to be in high pressure environments. They are in contact with many fluids, tissues, and cells of the body, and they have similar components and environments to wounds. With a lung fistula, we see that the hydrogel displays biocompatibility, biodegradability, and is bacteriostatic. Here is a picture of a lung fistula and the application of the hydrogel to stop the bleeding and air leaking from the opening. You can see the clear gel adhering quite nicely to the surface of the lung, which is in contact with blood and other fluids. Expanding on the hydrogel used for lung fistula repairs, we decided that the most effective bulk materials to use for our bioadhesive chitosan hydrogel would include chitosan, beta-glycerol phosphate, which is also known as GP, and gelatin. Now, chitosan is solubilized by aqueous solutions of acetic acid and lactic acid. It allows for cell adhesion, and we see that the greater the concentration of chitosan, the slower the rate of degradation. This allows us to form a biodegradable hydrogel that is also bactericidal and biocompatible. The GP, on the other hand, allows us to have greater control over the hydrogel by adjusting the pH to fit the conditions within the body. The GP also improves the elasticity of the gel to withstand high pressure environments and plays a role in the biodegradation of the hydrogel. We do want to ensure that a higher amount of GP is present with the chitosan to reduce the chemical changes due to temperature differences. Lastly, the gelatin is incorporated to add to the bioadhesive strength of the hydrogel and is the main component of our chitosan hydrogel. On the surface of our bioadhesive chitosan hydrogel, we decided that it would be extremely effective to incorporate antibiotics to fight infection immediately. Polydextrinaldehyde, or PDA, and polyethylamine, or PEI, are adhesives that kill gram-negative and gram-positive bacteria. They allow for the prevention of infections immediately, and in the wound dressing field, we have already seen its application in wound fillings. Prevention of infections are extremely hard to control, especially when on the battlefield. This application of the PDA and PEI to our chitosan hydrogel will save even more lives by helping to control and prevent infections. Going a bit further into the design concept of our chitosan hydrogel, we will see that it does indeed stop bleeding immediately at the site. The hydrogel acts as a plug until medical treatment can be provided within 4 hours of biodegradation. As stated earlier, the hydrogel is indeed biodegradable and allows for the prevention of infections due to not having to reopen or aggravate the wound to remove the biomaterial, which was seen in the XSTAT 30 wound dressing with the X-ray sensors within each sponge. This allows surgeons to immediately enter the wound and surgically repair the site of damage. With lower concentrations of the chitosan, but mixed with the GP, we are able to retain the strength of the chitosan to withstand the high pressure environment, while allowing it to be solubilized by aqueous solutions of acetic acid and lactic acid. As mentioned, the GP will be present in high concentrations and will make up most of the bulk of the hydrogel. For infection resistance, we decided to incorporate PDA and PEI to immediately combat infection. This will allow treatment to be able to be more focused on wound repair instead of infection control.
Toxicity issues. Improper aeration after ETO sterilization may lead to carcinogenicity in humans, which may contribute to the development of a number of different cancers. Although the cytostin hydrogel utilizes a plant-based cytostin in order to reduce potential for negative reaction, an inflammatory response may occur with the hydrogel when it is introduced to the wound depending on the person. Reaction to the polydendron aldehyde and the polyethanolamine adhesives. Sterilization options. Critical instruments/items may be stored in a way that maintain the integrity of a package and prevents contamination from any source. Items required to remain sterile must not be stored in ultraviolet cabinets or disinfectants, as these processes will compromise sterility. It is important that critical wrapped instruments are stored in a clean, dry area and are subjected to minimal handling before use. However, because this biomaterial will most likely be used among the military population and paramedics, it may be difficult to keep the material as clean as possible because of excess handling. Ethanol oxide gas is frequently used to sterilize materials that are otherwise too sensitive to heat or radiation sterilization. Many plastics fall into this category. Because of this, the plastic syringe that holds the bioadhesive cytosine hydrogel will be sterilized using ETO. Packaging requirements. Several essential performance characteristics of packaging include the ability to withstand physical conditions of specific sterilization processes, adequate air removal, sterilant penetration and contact with other contents, and eviction of sterilant, free of toxicity materials or dyes, and be low linting, adequate barrier to microorganisms and environmental contaminants, durability to resistant tears and punctures, and the removal of contents without contamination. We will design a disposable peel pouch. Disposable peel pouches are generally comprised of paper, plastic combinations, or Tyvek plastic combinations. Because Tyvek is intended for use in low temperature sterilization methods only, such as ethylene oxide and gas plasma, Tyvek will be the material of the packaging. The peel pouch will be designed to contain a lightweight item, which the syringe will be. Pouches must be sealed by heat sealing the open end or by using a self-sealing pouch with an adhesive strip that folded over the opening of the pouch. The package will be clear on one side and labeled on the other for easy identification. Peel pouches are advantageous when visibility of the content is important. It is important for those administering the gel that they can quickly identify the syringe and be able to open the package as quickly as possible because they will generally be administered during a time-sensitive period. Biocompatibility. Because the dressing can only be used up to 4 hours due to biodegrading of the material and because this is the maximum time body tissue can last without circulation, we will not need to worry about the material for long-term use. This material is intended to interact and form with the body in order to prevent further bleeding through biodegradation. The ability of this device to carry out its intended function for stopping bleeding and reducing infection with minimal interaction between device and blood that adversely affects device performance, without inducing uncontrolled activation of cellular or plasma protein cascades is the most important aspect of this material.
FDA issues. One issue surrounding preclinical and clinical trials is the long period of time required for testing if the FDA does not find the device to be substantially equivalent to an existing medical device. Hydrogels have been successfully marketed in the US, and they've also been used in the treatment of lung fistulas. Therefore, the approval process should be faster. Another issue is obtaining the proper patent and/or having to develop the proper syringe to fulfill the intended purpose. This would be the case if we were not using a syringe that is already on the market and is able to be used as intended. Time-consuming processes for FDA approval for animal testing are also an issue associated with the FDA. An additional issue is finding necessary participants for clinical research. This may require conducting research on specific populations such as military personnel. In addition, receiving the approval to be able to apply the hydrogel to all regions of the body may also be an issue. Unlike the XStat 30, which cannot be applied to the pelvis or above the collarbone. Preclinical trials, bench trials. Bench trials. These occur prior to testing in animals and in human subjects. They serve the function of eliminating mechanical flaws in the design of the device. An example of this may be improving the design of the applicator, and this may help with the ejection of the hydrogel itself. Bench trials are also used to observe the hemodynamics, wear, and durability of the medical device. Preclinical trials, animal testing. Animal testing occurs prior to testing in human subjects. It serves the function of assessing the safety of the device. Animal models mimic physiological functioning in humans, and this is why they are most useful. And for example, applying the hydrogel to an inflicted blast wound in a rat or another animal model may enhance researchers' understanding of how the hydrogel may perform in a human trauma wound.
In vitro testing. In vitro testing will be used for determining the possible effectiveness of various treatments, particularly antimicrobial and the healing-enhancing agents of the gel. It is difficult to maintain viability in organ cultures and to accurately test conditions because they vary from donor to donor. Additionally, modifications of the matrix or cellular composition is impossible with in vitro testing. To overcome these drawbacks, tissue explants from the same donor must be used for any given experimental series. This limits the number of samples and conditions a researcher can modify, resulting in limited data. Therefore, if positive results are seen through in vitro testing, in vivo testing will be next assigned. In vivo testing. All public health regulations will be followed and an institutional animal care and use committee will be utilized. Stress will be limited to prevent any adverse events from occurring. Rats will be the primary test subjects in order to reduce cost. A similar procedure will be used with the rats in this experiment as in one conducted by Buddy et al. To limit pain, on the day of injury, rats will be anesthetized with 4% isoflurane in oxygen and placed in a nose cone for continuous delivery of 1.5% isoflurane. To specifically target locations, a metal helmet will be placed over the rat's head in order to prevent head trauma during blast experiments because gel will not be effective for head trauma. At the completion of testing, rats will be euthanized with an overdose of pentobarbital that will be perfused through the left ventricle with 250 milliliters of phosphatate buffered saline, followed by 250 milliliters of 4% paraformaldehyde in PBS.
Clinical trials. Clinical trials assess the safety and efficacy of the medical device. They will help to observe how the hydrogel performs in a human subject. The testing of these clinical trials will likely occur in populations which are vulnerable to trauma wounds. Examples of these are military personnel. On a side note, although clinical trials are an essential part of understanding a medical device's safety and effectiveness prior to marketing, clinical trials may not be required for this medical device if the 510 clearance application is approved.
Surgical issues. The gel will be administered during a time-sensitive period, meaning there will be no time to sterilize the wound prior to administration of the hydrogel. In order to combat any infections that occur, antibiotics will be required pre and post-surgery. The interaction of the polydextrinaldehyde and the polyethylethamine with other antibiotics must be administered. Once the material is fully biodegraded, the surgeon must act swiftly in order to stop the bleeding and close the wound.
Issues unique to the device. One issue is the potential malfunction of the applicator. This would lead to an inability to administer the chitosan hydrogel. The applicator may not be able to hold the necessary amount of chitosan hydrogel needed to stop hemorrhaging in a large wound. At this point, we may need to resort to using more syringes. The cost of developing the device may also be high, which presents an issue with getting it to certain populations which may need it. Because this is an interim solution, the individual would be responsible for seeking additional medical treatment. If the individual is unable to receive the medical treatment in the appropriate time frame, the risk of heavy bleeding may be high. Potential risks associated with the device. Although biocompatible and biodegradable, the hydrogel may cause a reaction as a result of biological incompatibility or allergic reaction. Because the material is a hydrogel, removing the material may be difficult. Removal of the hydrogel by reopening the wound may also lend itself to risk of infection and hemorrhaging. This may also cause the individual unwarranted pain. Additionally, the spread of the chitosan hydrogel to various areas in the body may cause additional issues with clotting in areas where clotting is unwanted. Lack of blood flow to the affected area may lead to additional damage or decay. Marketing. Section 510K of the Food, Drug, and Cosmetic Act requires devices manufacturers who must register to notify the FDA of their intent to market a medical device at least 90 days in advance. This is known as the pre-market notification, also called PMN or 510K. This allows the FDA to determine whether the device is equivalent to a device already placed into one of the three classification categories. Because hydrogels have been successfully marketed in the US and used in the treatment of lung fistulas, our medical device may be considered substantially equivalent to an existing legally marketed device and may undergo a 510K clearance in order to avoid a prolonged pre-marketing process. Information which is to be included in the 510K clearance application includes, but is not limited to, classification of the newly developed device, predicate devices such as the hydrogels used in treating lung fistulas and sponges, final draft labeling, performance data, which includes bench trials, animal testing, and clinical trials, sterilization information, and guidance documents specific to newly developed device. Once the device is determined to be substantially equivalent to an existing legally marketed medical device by the FDA, the application submitter will receive an order stating that the device may be marketed in the US. At that time, the device is cleared for commercial distribution. The approval-refusal decisions are typically made within 90 days of submission of the 510K application to the FDA.